I'm going to try this again. It didn't quite work the first time. So I guess I had a practice run. So I'm going to run through meiosis real quick, drawing it for you out so that you can see it like the way I presented it to you in mitosis. So a couple of things about meiosis is that it only happens in reproductive structures or organs. For humans, that would be the ovaries and the testes. For flowering plants, it would be the stigma and the style. The stigma is what has the pollen, and the style is what has the egg. So meiosis only happens in reproductive structures. Mitosis, on the other hand, happens in what are called somatic cells or body cells. So mitosis happens in somatic cells. So here we go. We're going to start with meiosis, and my organism is going to have 4 total chromosomes. That means it has 2 pairs. And again, this is just fictitious for me to model meiosis for you. I can't draw 23 pairs in a likely time frame. And so the 2 types we're going to have are we're going to have big chromosomes and little chromosomes. Whoops. Just to make it easiest for it to draw, and if you didn't have colors to color them different colors, you could draw them as big and small. And so remember that half of your chromosomes come from your mom and half of your chromosomes come from your dad. And it should make sense to you after we get finished with meiosis how that happens. So one of the big chromosomes comes from the mother, and one comes from a father, little one from mom, a little one from dad. I should have put male and female. Yeah, other way around. Anyway, so you get one from one parent and one from the other parent. So here we go. Interphase happens, prophase happens, and remember in prophase you get the DNA condenses into chromatin. This is prophase 1. The membrane disappears. And so then we just truck along to the next phase. And that's metaphase. So remember in interphase that the DNA replicates. So I start with 4 here. I'm just going to go back to prophase, I guess. So we end up with this one copied itself to this one. This one copied itself to this one. This one copied itself to this one. So this is what we end up with. Same thing happens in mitosis. That's what we end up with also. But you have a new term that you got introduced to called homologous chromosomes. And those are chromosomes that are the same size and carry the same genetic information. Genes. So this is a set of homologous chromosomes. This is a set of homologous chromosomes together. So in mitosis, we talked about the sister chromatids. In meiosis, we're going to add a layer, and we're going to talk about homologous chromosomes. So we have the large chromosome and the large chromosome together. Both of these together are homologous chromosomes. The little ones together are homologous chromosomes. Remember that homo means the same. So prophase 1. Now we go to metaphase 1. Now this is going to be different than it was in interphase. In metaphase 1, the homologous chromosomes line up at the equator. So homologous chromosomes, homologous chromosomes. Are there still sister chromatids? Yes. This is a sister chromatid to this one. This is a sister chromatid to this one. But in metaphase 1, it's the homologous chromosomes that line up and not the sister chromatids. So remember in mitosis, to compare, we had the sister chromatids that lined up at the equator. But in metaphase 1, it's the homologous chromosomes that line up. So that's one difference. Then we go to anaphase 1. And in anaphase 1, the sister chromatids were broken apart, and one sister chromatid was pulled to the poles. In anaphase 2, the complete homologous chromosome gets pulled to the poles. So they don't get separated, the chromatids here. They stay together. So you need to be able to contrast the two processes. Okay, so then mitosis ends up here with two cells. They're genetically the same as the beginning cell. Here's where we start the second cell division of meiosis. There's no DNA replication here. The only place that DNA replicates in our processes is here in the beginning. So it only happens here at the beginning in both processes. So in prophase 1, I'm sorry I didn't draw your two cells here. Now we go to prophase 2, and we have our two cells from the first division, and the DNA condenses, the nuclear membrane disintegrates, all that other normal good stuff. Nope, not the right drawing. Eraser, eraser. So we have our homologous, one of our homologous chromosomes. Okay, so now here we go with metaphase 2. And here we have the chromatids line up. So that's one cell. And there's the other cell. Then we go to anaphase. And then we go to our telophase. Lucy. So at the end of meiosis, we have cells that are not genetically identical. To the mother cell. Lucy. We have 2 cell divisions. And we end up with 4 cells. In mitosis, we end up with identical daughter cells. One cell division and 2 cells. Meiosis only occurs in reproductive structures. And mitosis occurs in all the other cells, in the somatic cells. So if we compare the cells that we end up with, in mitosis, our ending daughter cells have 2 of each type of chromosome. In meiosis, we only have 1 of each type. So 2 of the possibilities came from the female, and 2 of the genetic possibilities came from the male.